scarcity of food due to natural calamities like floods, and or the person cannot buy food due to extreme poverty, or involuntarily due to the desire to lose some weight, or it can be due to hunger strikes or clinical conditions like major surgery. So the reason can be anything, but if there is no glucose coming in from the gut, if no food is being taken by the mouth and none of the nutrients are being absorbed, it is termed as starvation. So, in the starvation state, the body is deprived of carbohydrates, lipids, proteins, vitamins, minerals, everything. The calorie providing substances like carbohydrates and proteins are also deficient. The building blocks proteins are also deficient, and the growth factors are also not there, the vitamins and the minerals. So, during the emergent starvation, the body is trying to get adapted to and manage with the endogenous fuel reserve. So there is something in the body itself, there is fuel stored in the body itself, which the body tries to mobilize and sustain itself. So do you remember any such fuels? One is the glycogen, and the other is the triacylglycerol reserve, and the body can also undergo gluconeogenesis, that is make new glucose molecules. So there is some stuff within the body itself which the body mobilizes in order to derive energy, because there is no intake of food, nothing is coming in, nothing is getting eaten and nothing is getting absorbed, but the body has to do carry out the functions. There's work is being done and the ATP requirement is there, which the body tries to fulfill by mobilizing the endogenous fuel reserve. So in the absence of food, plasma levels of glucose, amino acids, and triacylglycerol falls, and this triggers a decline in insulin secretion and increase in glucagon and epinephrine release. So the decrease in the level of insulin and the increase in the level of counter-regulatory hormones like insulin is there, and there is also decreased availability of substrates. So this makes the period of starvation a catabolic period. Catabolism is going on. What is catabolism? In this period, the body store of triacylglycerol, glycogen, and protein are degraded to generate the energy. Catabolism means the stored substances, the fuel reserve are getting used up. The stored substances like triacylglycerol is getting broken down, it is getting released from the white adipose tissue, it is getting degraded to free fatty acids and glycerol, and the free fatty acids are oxidized for energy. So all the stored substances are getting mobilized in the period of starvation. So this shows the metabolic fuel reserve present in a 70 kg man at the beginning of a fast. In a normal 70 kg man, there is 15 kilogram of fat, which gives around 135,000 kilocalorie of energy. There is 6 kilogram of protein, which gives 24,000 kilocalories of energy, and there is zero point 0.2 kg glycogen, which gives 800 kilocal energy. So this is the fuel reserve present. This is for an average man, 70 kg man. Although here the protein is listed as a source of energy, all the proteins they cannot be used because uh, it is required for other functions as well. So you can see from this chart that the mean stored reserve is provided by the fat, which is triacylglycerol. This is the mean energy reserve, and little amount is glycogen. So in the period of starvation, all these stored substances are mobilized to make ATP so that the body can function. So there are different metabolic pathways that are taking place during starvation. Firstly, you need to understand that there are some organs and tissues which can only utilize glucose for energy, like RBCs. They cannot utilize any other fuel. They cannot utilize the free fatty acids. They cannot utilize the ketone bodies. So the only fuel for the RBCs is the glucose. So these must be provided with glucose. So there are two processes that are going on to ensure that the glucose requiring tissues are getting enough amount of glucose. Firstly, there is glycogenolysis. What they mean by glycogenolysis means the breakdown or degradation of the stored glycogen. Is it much? Is the glycogen present in a large amount? No, I just showed you the fuel reserve. Glycogen provides only about 800 calories. That's it. So this stored glycogen, it finishes in about 10 to 18 hours. So after that, what process makes sure that there is enough glucose? It is the process of gluconeogenesis. Gluconeogenesis means there is production of new molecules of glucose from non-carbohydrate sources. Using non-carbohydrates, the liver makes new molecules of glucose for the glucose requiring tissues. It is mainly the RBCs because RBCs cannot use any other fuel molecules other than glucose. And then there is also brain, which prefers to use glucose as long as it is available. But I hope you remember that there is another fuel that the brain can use, and they are the ketone bodies. So, okay, for talking about the glucose, these two processes they make sure that the glucose requiring tissues get enough amount of glucose. Glycogenolysis for a short period of time, and then the level of glucose is maintained by gluconeogenesis. And then there is another process going on, and that is mobilization of fatty acids from the adipose tissue. The triacylglycerol, which is present in a high amount, which gives a lot of calories. So that is mobilized from the adipose tissue, and most of the organs can switch to fatty acid to using fatty acids as alternate fuel during starvation. And then there is third process, third fuel mentioned, that is since an increase of ketone body on the liver supply energy. So when there is sufficient amount of ketone body in the blood, the brain also starts to utilize the ketone bodies. The first process that starts is glycogenolysis. During the first few hours after meal, the tissues use glucose, but several hours after a meal, the blood glucose levels it starts to decline. So there is increased secretion of glucagon and decrease in the level of insulin. There is mobilization of liver glycogen store. So when as the glucose level decreases, to prevent hypoglycemia, liver releases glucose in the bloodstream. The liver glycogen is broken down and glucose is released in the bloodstream. But the liver glycogen store is small, only 100 gram. So this store it lasts for only about 10 to 18 hours of fasting. This is the transient response to early fasting. So beyond that, there is no liver glycogen to supply glucose or to maintain the blood glucose level. So, what are the different sites that glycogen is stored in? One is the liver, and the other is the muscle. Only about 100 gram of glycogen is stored in the liver, but in the skeletal muscle, there is about 400 gram of glycogen. But this glycogen stored in the muscle, it does not contribute to blood glucose directly due to absence of the enzyme glucose 6 phosphatase. To convert the glycogen into glucose, in the last step you need glucose 6 phosphatase. But in the muscles, this enzyme is absent, so glucose cannot be released in the blood. So you can see, even think of muscle as being selfish because it only keeps its glycogen for itself and does not release the glucose to maintain the blood glucose level and for other tissues to utilize it. So this is useless to maintain the blood glucose level. Only the liver glycogen helps in the maintenance of blood glucose level. The first thing that is going on during starvation or even early fasting is there is glycogenolysis going on. And there is second fuel which is present in a large amount, and that is the triacylglycerol. There is fatty acids stored in white adipose tissues. In the state of starvation, there is increased degradation of fat and release of free fatty acids. What happens to these free fatty acids that are getting released? They undergo beta oxidation. I hope you remember the process of beta oxidation, the steps of beta oxidation, the energetics of beta oxidation, how many ATPs are getting produced by one beta oxidation of a single fatty acid. Beta oxidation takes place, which leads to production of a lot of ATPs. So, do you remember this? There is increased release of fatty acid. Why? How are these fatty acids obtained? Fatty acids are obtained from hydrolysis of stored triacylglycerol. So we have the triacylglycerol in the adipose tissue. There is this enzyme, hormone sensitive lipase, which is activated by glucagon in the state of starvation, which is a catabolic state. Glucagon is high, due to which hormone sensitive lipase is activated. Hormone sensitive lipase degrades triacylglycerol to diacylglycerol, and this diacylglycerol is acted upon by other lipases, and you get glycerol and free fatty acids. The free fatty acids, they exit the adipose tissue, they are released in the blood. In the blood, the free fatty acid, they bind to the albumin present and are transported to tissues, and they enter the cells. The fatty acids are converted to their active form, will undergo beta oxidation, and you get acetyl CoA. You've already studied these steps. You've already studied how lipolysis takes place and how beta oxidation takes place. So, as I said, in the state of starvation, glucagon is high in amount, epinephrine is high, and insulin is low. This enzyme, the hormone sensitive lipase, is active in its phosphorylated state and inactive in its dephosphorylated state. When glucagon is high, the hormone sensitive lipase is in phosphorylated state. And when insulin is high, hormone sensitive lipase is in dephosphorylated state. In the state of starvation, glucagon is high, epinephrine is high, due to which hormone sensitive lipase is phosphorylated and active. So you can say that glucagon activates lipolysis in adipose tissue by making hormone sensitive lipase active. You don't need to make this figure. This is just for a recollection of what was taught in the first year. So in the state of starvation, the triacylglycerol store is degraded because the hormone sensitive lipase is active when glucagon is high. And there is increase in fatty acid oxidation as well. So, do you remember about the carnitine shuttle? So, do you remember this enzyme, acetyl-CoA carboxylase?
Yes, ma'am.
Yes, ma'am. Yes. What is in which synthetic pathway is this acetyl-CoA carboxylase regulating enzyme? Acetyl-CoA carboxylase
Fatty acid synthesis.
in fatty acid synthesis. Okay, good. In the fatty acid synthesis, acetyl-CoA carboxylase is active. But how is this acetyl-CoA carboxylase regulated? Insulin makes it active and glucagon makes it inactive. So, in this case, in the case of starvation, when glucagon and epinephrine are high, acetyl-CoA carboxylase is present in its phosphorylated form, which is the inactivated form. So, fatty acid synthesis is not going on. Acetyl-CoA carboxylase is inactive. Now, look at this step. This is the first reaction in the synthesis of fatty acid. Acetyl-CoA is getting converted to malonyl-CoA. The enzyme is acetyl-CoA carboxylase. Carbon from carbon dioxide is getting added to acetyl-CoA and you're getting 3-carbon malonyl-CoA. 1 ATP is utilized for this process. This is the first step in the fatty acid synthesis. So, when fatty acid synthesis is going on, lot of malonyl CoA will get formed. But when fatty acid synthesis is not going on, the level of malonyl CoA is low. Understood? So, so do you remember how carnitine shuttle is uh, the carnitine acyltransferase 1 enzyme of the carnitine shuttle, how is it inactivated? What inhibits it?
Malonyl
Malonyl
CoA.
coenzyme is the inhibitor of the carnitine acyltransferase 1 or enzyme. So, during, so in this state, in the state of starvation, when malonyl CoA is low, when acetyl CoA is not getting converted to malonyl CoA because acetyl CoA carboxylase is inactive. When malonyl CoA is low, it does not inhibit the enzyme carnitine acyltransferase 1. So when carnitine acyltransferase 1 is not inhibited, the free fatty acids which you got from the lipolysis, they enter the mitochondrial matrix and they undergo beta oxidation. The fall in malonyl CoA due to phosphorylation of acetyl-CoA carboxylase, meaning due to inhibition of acetyl-CoA carboxylase, removes the brake on carnitine acyltransferase 1, which allows the beta oxidation to take place. So, a lot of things that we talk about today, uh, these processes you have studied in your first year under cardiovascular system. If you remember those, then it is very easy for you. If you do not remember it, today when I'm talking about this. If you're getting confused, you please go back and you revise what was taught there, because you need to remember what was taught in your first year to understand all these. So at the end of beta oxidation, you get acetyl CoA, which goes to the Krebs cycle, and you get NADH, and you get FADH2. So from all these, you get a lot of ATPs. I hope you remember how many, because it can be asked in your Viva. When lipolysis was going on, when the triacylglycerol was getting degraded, you got glycerol and three free fatty acids. The free fatty acids go beta oxidation. What do you think happens to the glycerol? Is it wasted? No. The glycerol that is derived from lipolysis is used as a substrate for gluconeogenesis, that is making of new molecules of glucose from non-carbohydrate sources. The body uses glycerol that it got from lipolysis to make new molecules of glucose, so that the glucose requirement of the tissues that need glucose is fulfilled. But you keep in mind that the amount of glycerol that you get from lipolysis is not enough to generate the glucose that is needed by the brain and the other tissues that can only utilize glucose. So, the process doing gluconeogenesis only from glycerol derived from lipolysis is not enough to make the necessary amount of glucose. So you need some other source of glucose. Uh, the from the gluconeogenesis from glycerol only gives you about 30 gram of glucose per day. Only 30 gram. But the brain needs around 120 gram of glucose per day. So there is a deficient. Only 30 gram of glucose is getting made from the glycerol, gluconeogenesis from glycerol, but you need 120 grams. So there is around 90 gram per day of glucose that is deficient. So do you think there is any other substrate for gluconeogenesis? Yes, there is. Another substrate for gluconeogenesis is the muscle proteins. There is proteolysis going on. You've got the muscle protein, which can be the substrate for gluconeogenesis. There is proteolysis going on. This process of proteolysis, it is initiated by fall in the level of insulin. In this state of starvation, insulin is low, which initiates the process of proteolysis. So during the first few days of starvation, there is rapid breakdown of muscle protein because you need the substrate for gluconeogenesis. This process of breakdown of muscle protein gives you the amino acids that are used by the liver for gluconeogenesis, because as I said, only the glycerol from lipolysis is not enough to meet the needs of the brain and the other glucose requiring tissues. So the body needs to break down the muscle protein. So the main amino acids are alanine and glutamine, which are the most important gluconeogenic amino acids released from the muscle. So these amino acids, they are made into new molecules of glucose, which can be used by the brain and other glucose requiring tissue. But do you think the process of proteolysis, is good for the body? No. This is putting a huge strain on the level of the protein. This is not good because the proteins have other function as well. You do not want all the muscle to be broken down to be made into new glucose. Look at this. Gluconeogenesis is synthesis of glucose from non-carbohydrate precursors. The site of gluconeogenesis is liver mainly and later on kidney. The substrates for gluconeogenesis are the glucogenic amino acids and glycerol from adipose tissue. Only gluconeogenesis from glycerol is not adequate. That is why there is muscle proteolysis going on to give the substrate for gluconeogenesis. This process of gluconeogenesis, it begins 4 to 6 hours after last meal and it becomes fully active as the liver glycogen depletes. Some tissues like brains, RBCs, kidney medulla, lens, testes, they require a continuous supply of glucose as a metabolic fuel. They need the glucose. So, as the liver glycogen can only meet the needs of all these tissues for about 10 to 18 hours, in the absence of dietary intake of carbohydrate, there is need for gluconeogenesis. So this new glucose molecules are getting made for the glucose requiring tissues. So do you think these glucose are also used by the muscles? No, the muscles they do not use this glucose because the glucose in transport into the skeletal muscle is dependent on the insulin sensitive GLUT4 transporters. These are insulin sensitive. In this state there is low level of insulin, so that is why uptake of glucose, the glucose transport into the skeletal muscle cell and subsequent glucose metabolism is depressed due to the low levels of circulating insulin. So the glucose that we get from the hepatic gluconeogenesis is not utilized by the muscles and the adipose tissues because the transporters that they use to uptake the glucose, the GLUT4 is insulin sensitive. So the glucose that the liver is making by the process of gluconeogenesis is utilized only by the glucose requiring tissues, the tissues that can only utilize glucose as fuel, and by the brain which prefers to use the glucose. But this process of gluconeogenesis is putting a huge strain on the muscle proteins. So to prevent that, the body comes up with another product or another molecules that it can use, and it is the ketone bodies. The blood glucose is maintained by hepatic gluconeogenesis from glucogenic precursors like amino acids and glycerol. But in the prolonged fasting, there is production of ketone bodies as well. So, here, let's read this. In the early days of fasting, the brain uses only glucose as fuel. I said before that the brain requires about 120 grams of glucose per day. So to make that glucose by the process of hepatic gluconeogenesis, this process is putting a huge strain on the muscle protein because you need the substrate amino acid and you get it by the proteolysis. And there is also another substrate which gives around 30 grams of glucose per day, and it is the glycerol from lipolysis. So these two substrates are getting used to make the glucose for utilization by the brain and other glucose requiring tissue. Glucose needed by the brain is in huge amount. So to prevent the strain on the muscle protein, to protect the muscles from excessive proteolysis, ketone bodies are made. ketone bodies can reduce the brain glucose use. So as the level of ketone bodies in the blood is built up, the brain also starts to use the ketone bodies. So when brain starts to use the ketone bodies, the glucose need by the brain is decreased. When the glucose need by the brain is decreased, there is reduction in the need for protein catabolism for gluconeogenesis. So if you do not need a lot of glucose for the brain, then excessive proteolysis is not required. The muscle proteins are preserved. So when the brain starts to use the ketone bodies, only 40 gram of glucose is then needed per day for the brain, compared with about 120 gram in the first day of starvation. So when the ketone bodies are made and when the brain utilizes the ketone bodies, the glucose need of the brain reduces from 120 gram to around 40 gram per day. So this is almost fulfilled by the gluconeogenesis from only the glycerol. So the muscle proteolysis is minimized. The muscle proteolysis is almost stopped because you no longer need the amino acids for gluconeogenesis. So this way, after the generation of ketone bodies, the muscle proteins are preserved. So this has been covered in your first year, but again, let's go through it. So this is the adipose tissue, this is the liver. Let's suppose this is the liver. So you have triacylglycerol stored in the adipose tissue. This undergoes lipolysis and you get glycerol and free fatty acids. The glycerol is released in the blood, goes to the liver, where it is utilized as substrate for gluconeogenesis and you get new molecules of glucose. The fatty acids, they are bound to albumin and they are carried in the blood and they reach different sites. Now let's talk about what happens in the liver only. And they also reach the liver. In the liver, these free fatty acids, they undergo beta oxidation and you get acetyl CoA, FADH2 and NADH. So this acetyl CoA, it can either enter the citric acid cycle or this same acetyl CoA can enter the pathway of ketogenesis. So when does acetyl CoA enter the citric acid cycle and when it goes towards the ketogenesis? So when the citric acid cycle is not functioning because oxaloacetate is deficient, then acetyl CoA is directed towards the ketone body synthesis. Ketogenesis is favored when the concentration of acetyl CoA produced from fatty acid metabolism exceeds the oxidative capacity of the TCA cycle. Why this acetyl CoA exceeds the oxidative capacity of TCA cycle? Because in the first step, there is condensation of acetyl CoA with oxaloacetate. I hope you remember the TCA cycle. Acetyl CoA condenses with oxaloacetate, but in case of starvation, that oxaloacetate is also diverted towards the pathway of gluconeogenesis. So this acetyl CoA, it is diverted towards the pathway of ketone body synthesis. So you don't have to write this when you talk about the biochemistry of starvation. This is just for your understanding. Now let's talk about the role of kidneys. In the later phase of starvation, around 50% of gluconeogenesis occurs in the kidneys. Initially, only gluconeogenesis occurs in the liver, and later on, 50% occurs in the kidneys. The kidney also provides compensation for the acidosis that accompanies the increased production of ketone bodies. So the glutamine that is released from the muscle metabolism of branched chain amino acid. So the muscles are getting degraded. Branched amino acids are released. So the glutamine is getting released from the muscle metabolism of branched amino acid. This glutamine is taken up by the kidney and acted upon by the renal glutaminase and glutamate dehydrogenase. So this process, when glutamine is acted upon by glutaminase and glutamate dehydrogenase, this produces alpha ketoglutarate. This alpha ketoglutarate can be used as a substrate for gluconeogenesis, and there is also ammonia that is getting produced. This ammonia, it picks up the proton, the H plus, from the ketone body dissociation. Ketone bodies are acidic in nature. They can release the proton. They decrease the pH of the blood. Ketoacidosis ones. I mean, these are acids which decrease the pH of the blood. So this proton is getting released from the ketone body dissociation, which is picked up by the ammonia, and it is excreted in the urine as ammonium ion. So this process decreases the acid load in the body. So this is the role of kidneys in starvation. It maintains the it provides compensation for the acidosis which can occur due to overproduction of the ketone bodies. Now look at this figure. It shows the interrelation of different organs in starvation. This is the liver, brain, adipose tissue, and the muscles. So from the pancreas, glucagon is released, level of insulin is decreased, due to which all these processes are going on. There is gluconeogenesis going on, there is ketone body synthesis going on. The brain, it initially it only utilizes glucose, but when there is adequate level of ketone bodies present, it starts to utilize the ketone bodies, thereby decreasing the glucose requirement, which leads to decrease in the process of proteolysis, thus preserving the muscle proteins. So initially proteins are present on the muscles are degraded, and you get amino acid, glutamine, and alanine, which is used for the process of gluconeogenesis to make new molecules of glucose. From the triacylglycerol present in the adipose tissue is degraded, and you get free fatty acids and glycerol. Glycerol is used as a substrate for gluconeogenesis, whereas these fatty acids, they undergo beta oxidation, and you get acetyl CoA. The acetyl CoA can either be oxidized in the TCA cycle to give you ATPs, or it can be used for the synthesis of ketone bodies, which can be utilized by the muscles, it can be utilized by the brain. So this figure shows the interrelation of different tissues, organs during starvation. Do you have the book Lippincott? The figure given in the Lippincott about the interrelationship of different organs during starvation is clear, easy to remember, and in your exam sometimes you might be asked to give the diagrammatic representation of the metabolic changes during starvation. So I want you to all to go through the figure that is given. You can either make this one, or the figure given in Lippincott is better. You can go through that figure and practice making it, because you can sometimes be asked to give the diagrammatic representation. So please practice it. So this is the summary of all the steps that is going on during starvation. The first priority of the body is to provide sufficient glucose to the brain and other tissues that are dependent on it. So, how does this occur? Firstly, there is glycogenolysis going on in the liver, which maintains the blood glucose level for 10 to 18 hours. And then gluconeogenesis starts by the liver, which maintains the blood glucose level. There is also mobilization of triacylglycerol stored in the adipose tissue, and the muscles shift from glucose to fatty acid use as fuel. There is proteolysis going on, which provides carbon skeleton for gluconeogenesis, and in the first few days, there is rapid breakdown of the muscle proteins. So, this is what is going on during starvation. The first priority of the body is to make sure that the glucose requirement by the brain and the other tissues are met. And the second priority is to preserve the muscle proteins. After 3 days of starvation, liver forms a large amount of ketone bodies and releases it into the blood. When this happens, there is shift from utilization of glucose to utilization of fatty acids and ketone bodies. After several days of starvation, the major fuel for brain is ketone bodies, as a result of which gluconeogenesis decreases and less muscle is degraded than in the first days of starvation. So, the production of ketone bodies leads to preservation of the muscle proteins. So, for the generation of ketone bodies, you need acetyl CoA, which is coming from the beta oxidation of fatty acids. Those fatty acids are coming from the breakdown of triacylglycerol depot. So you can say that a person's survival time is mainly determined by the size of their triacylglycerol store. So if you have more triacylglycerol, then you can survive for a longer period of time. So what do you think happens after depletion of the triacylglycerol store? When the glycogen is finished at the earliest 10 to 18 hours. After that, the triacylglycerols get finished. When there is depletion of the triacylglycerol stores, the only source of fuel that remains is protein. Protein degradation, it accelerates, and death results from the loss of organ function, heart, liver, or kidney. It stops working. So that is when a person dies. So this is it about starvation. You learn these two slides properly. All the processes are written here, and you practice making the figure that is given in Lippincott. I hope you understood today's class. Uh, in the